Okay, so welcome everyone to the last session of Getting into the Weeds for 2024. This session is going to be on cultural practices to improve disease control. And without too much further ado, Dustin is going to take the reins in a second, and he's going to guide us through cultural practices to improve disease control on your operation.
So today I'm going to talk about cultural practices to improve disease control. So an introduction. We'll be going through a number of practices that you can use on farm to aid you in controlling disease outbreaks. So these practices will be paired with examples of actual crop disease interactions to help you make informed decisions regarding crop rotation, sanitation, and other control options. So certain cultural practices are invaluable in reducing plant disease losses. A control program is enhanced whenever you can utilize as many methods of control as possible. So new strains of an organism may develop that will attack resistant varieties or becomes tolerant to certain pesticides over time when these practices are used alone. So combining practices reduces the risk of failure. So the term cultural control describes the activities of humans aimed at controlling disease through the cultural manipulation of plants. For some crops, such control practices may be the only economically viable method available. Many cultural practices were features of traditional farming and have been replaced as cropping has become more intensive. High labor costs in modern agriculture may prohibit the use of many of these practices. Cultural control practices could be considered a form of biological control and tend to be preventative and indirect in their actions against pathogens. So the success of cultural control practices ultimately depend on understanding the biology of the pathogen and the response of the host to infection. Such knowledge facilitates management decisions which attack parasites at vulnerable stages of their life cycles. It may be difficult to assess their effectiveness sometimes because the farming operations are often performed well in advance of the expected pathogen attack. Historical evidence indicates that where pathogens have been introduced into previously clean sites, disease epidemics usually follow shortly after. The spread of disease to new areas, new plants, or new plant products can be prevented or reduced by incorporating quarantine and sanitation practices. First thing we're going to talk about is site selection. So a grower may incur significant losses if susceptible crops are planted in fields known for having a history of soil pathogens. Plant pathogenic fungi such as armillaria, Fusarium, Plasmidiophora, Sclerotiarum, and Verticillium are true soil inhabitants and will persist in soil for many years, even in the absence of a plant host. However, soil-borne fungi such as Phytophthora, Pythium, and Rhizoctonia often are much more widespread. Site selection, unfortunately for these, may be less of an option in avoiding these pathogens. If possible, you can avoid planting crops in low-lying wet areas given the higher incidence of soil-borne diseases. So in choosing a site for a crop, it's wise to check the previous cropping history of the land to ensure the pathogen that may be present from a previous crop will not infect the crop to be planted. Selection of resistant cultivars. So the use of resistant cultivars is one of the most important and economical components of an integrated disease management program. Resistant cultivars offer one of the most successful approaches to the control of pathogens of many crops, especially those diseases that cannot be controlled by other means. The term resistance usually describes the plant host's ability to suppress the activity and progress of a pathogen, which results in the absence or at the very least reduction of symptoms. So a critical decision in crop production is the selection of the cultivars to be grown. So some cultivars are resistant to particular pathogens and are inherently less damaged than other genetically related plants growing in the same location. Resistant cultivars have provided one of the most successful approaches to the control of pathogens of many crops, especially those which cannot be controlled by other means. However, continuous plant breeding programs are often necessary to overcome the effects of the development of new races of pathogens and breakdown of host resistance. Breakdown of host resistance can happen over time as pathogens mutate. So you can have a variety that is initially very resistant to a specific pathogen species, and over time, through recombination, through other events, that pathogen can actually mutate and become it can become more virulent or more aggressive, and the plant that previously had resistance to that pathogen no longer does. So growers therefore need to be aware of the availability of new cultivars and be able to evaluate their potential in relation to their other agronomic characteristics. So planting pathogen-free and high-quality seed. So planting pathogen-free and high-quality seed is a critical first step in managing disease. So this is particularly important for vegetatively propagated material, such as tubers and bulbs. True seed as well can also contain pathogens, however, and all can serve as the source of entry of pathogens into new areas. So all of the major plant pathogens can be transmitted by seed or propagating material. Materials which have been certified free from disease following field inspection or laboratory testing provide a useful safeguard to growers. So I'll plug right now. We have the Plant Health Lab, which is available through Perennia, where we're actually able to test for a number of these plant pathogens. So that's a service people can look into if you're unsure of whether your material contains pathogens or not, fungal or bacterial pathogens. You can send them into the Plant Health Lab to test. We're actually able to test pathogen presence on seeds as well. So the value of certified seed is further enhanced if the parent crops have been grown in an area where particular pathogens are unknown. Alternatively, rigorous chemical control programs for selected organisms may be instituted during crop growth and storage to reduce or eliminate pathogens from planting materials. If these procedures are not followed, growers may have to treat seed or other propagating structures prior to sowing. Efficient seed cleaning procedures also contribute to the control of some diseases by removing infected seeds and leaving only uninfected material. Pathogens can be transmitted to healthy material by contaminated tools as well during the preparation of vegetatively propagated material for planting. In such situations, growers need to practice proper hygiene procedures to prevent transmission of pathogens. So make sure that tools you're using are cleaned and sterilized in between plantings. So for vegetatively propagated crops, so things like carnations and chrysanthemums, for instance, tissue culture techniques may be necessary to obtain disease-free planting stock. So for example, virus and vascular wilt diseases such as Fusarium and Verticillium, they don't actually reach the apical meristems of their hosts until late in the development of the disease. So pathogen-free meristems can be tissue cultured and used as the basis of a vegetative propagation program. Even if planting materials are apparently free from disease, infection may occur if they are damaged during planting operations. So some common causes of damage include the incorrect adjustment of mechanical planting devices and seedling injury that occurs during transplanting. There's also rotation with unrelated crops, which is probably the most utilized cultural practice for disease control. So one should not expect rotation to eliminate disease development, but it certainly aids in reducing damage from most diseases. So some pathogens that cause disease survive in the soil from year to year in one form or the other, usually as sclerotia, spores, or hyphae. So they have survival apparatus that they can survive in the soil. So continuously cropping the same crop builds up the population levels of any soil-borne pathogen of that crop that may be present. So the population can eventually reach a level that's so large that it becomes difficult to grow the crop without yield losses. So rotations are most likely to be effective in controlling pathogens which only survive in the presence of a specific host or its residues. That way, you can rotate from a crop that may be, say, susceptible to that pathogen that's to one that's not susceptible. So this is particularly useful in species such as downy mildew and powdery mildew, where there's specific hosts. So a powdery mildew of cucurbits, say, does not potentially doesn't affect cereals. Like there's a powdery mildew of cereals as well, and each crop. But when you're dealing with things that are generalists, so a lot of soil-borne pathogens like Pythium and Phytophthora, sometimes it's or particularly Verticillium, it's difficult to rotate into a crop species that may not be a host for this pathogen. So for instance, Verticillium infects 200 different plant species. So trying to find, if you have Verticillium and trying to rotate into something that isn't susceptible to Verticillium can be very, very difficult. So some rotations may include a crop which inhibits the development of a specific pathogen. So high levels of root knot nematode, for instance, occur in soils following crops such as field and sweet corn. However, other crops such as forage sorghum, they host very few nematodes. So crops of susceptible plants following these crops show little sign of nematode damage. So it actually breaks up the nematode population. Legumes have also been used to reduce populations of the nematode Pratylancus in soils. Similarly, marigolds inhibit the development of a range of nematode species, and many brassica crops produce glucosinolates, a chemical that actually breaks down to produce another chemical called isothiocyanates, which are active against a number of soil-borne pathogens. So those can be effective control methods as well. So by rotating crops, soil-borne pathogens will eventually decline without a suitable host. So the most successful rotations employ intervals between susceptible crops, which are longer than the known survival period of pathogens. So in most instances, crop rotations are usually for a definite time period of around 2 to 3 years. A longer time period of 4 to 6 years may be required for control of some diseases, especially on brassicas and potatoes. So when you're dealing with pathogens that produce very resilient spores that are able to survive in the soil, so things like Pythium, as well as if anyone's familiar with clubroot, Plasmodiaphora, they're capable of surviving sometimes in the soil for up to 20 years. So then you're dealing with a significantly longer rotation. So the crop rotation may also include a fallow period in which land is rested from production as well. So probably the most widely used rotations are to control soil-borne pathogens of cereals. These often include either a non-host legume crop, which can improve soil fertility, as well as reducing inoculum, or a fallow period during which alternate weed hosts and volunteer plants are removed or destroyed. So take all, foot rot, ascochyta, and some other pathogens of wheat have been controlled in some regions by removing grass hosts, especially during fallow periods. Long-term rotations are required to control pathogens such as Streptomyces scabies, which is common scab of bacteria, and Rhizoctonia solani, which can survive in the soil as resistant spores or as sclerotia. So this is a photo I wanted to show. When you're rotating, you really need to be cognizant of the disease life cycle of the pathogen, particularly because if you're not familiar with fungi, they always have two names, which can sometimes be confusing as well. Taxonomists tend to change names every 5 years or so, which also makes it difficult. But in general, they give fungi two names. So they give the name of the asexual stage and they give the name of the sexual stage. So for instance, if you plant wheat and you get Fusarium head blight, for instance, which is caused by Fusarium graminearum, and then you decide, okay, I need to rotate out of my wheat, I'm going to plant corn. You look it up and you say, does corn have get Fusarium head blight? And you don't find anything. No, there's no instance of Fusarium in corn. That's great. The problem is that corn is susceptible to Fusarium's sexual stage, which has a different name, Gibberella zeae. So sometimes it's important to get that information, particularly we're dealing with a fungus, its complete life cycle. So sometimes the life cycle isn't always known. So there'll only be a sexual stage or an asexual stage, and that they're not aware of the other. But when you're looking up fungal pathogens, particularly, always try to keep that in mind, that there's an asexual stage and a sexual stage, and they may infect different crops. So it's very important that you're aware of this complete life cycle, because you don't want to plant wheat and then rotate into corn and you're just building up that entire Fusarium gibberella life cycle. You're actually not decreasing that population whatsoever. So the next is physical removal and destruction of crop residue. So crop residues provide suitable substrate for many pathogens. Physically removing and destroying, so burning and burying crop residues, are important cultural control practices performed during intercrop periods. So the effect of destroying crop residue on particular pathogens depends on the type of crop, the extent of the cropping area, and the survival mechanism and host ranges of the target pathogen. So burning crop residue generally has been discouraged because of destruction of valuable organic matter and the creation of air pollution. However, it is highly effective means of eradicating some disease-causing organisms associated with crop residue. So many pathogens are eliminated by the intense heat produced from burning. So where this is practice does not occur, post-harvest burning of trash can be employed as a disease control practice as well. So the success of burning and destroying inoculum is determined by a few factors. So the intensity of the burn, the amount, moisture, content, and quality of the plant debris, the wind velocity, and the duration of temperature of the burn. However, any advantages in disease control has to be weighed, like I said, against the adverse effects of burning. So these include loss of nutrients, contamination of the environment with smoke, addition of carbon dioxide into the atmosphere, contributing to global warming, soil erosion following the removal of protective vegetation as well. Removing diseased plants and plant parts. To eliminate sources of inoculum is carried out routinely in many nurseries, greenhouses, and high-value crops. So for instance, strawberry leaves infected by various leaf-spotting fungi are removed early in the season. Diseased parts of larger plants such as tree crops are often removed by pruning. So for instance, infected branches are removed in cases of fire blight, Erwinia mellivora, certain apple cankers like Nectria, powdery mildew of apples, and silver leaf of plums. Mummified fruits as well as diseased twigs should be removed from palm and stone fruits to reduce the inoculum source. Subsequent sanitation measures usually involve burning pruned material and treating cut surfaces with suitable fungicides or other chemicals. Pruning inevitably influences the microclimate within the tree canopy, indirectly affecting any subsequent disease development. In mixed farming enterprises, livestock could be used to graze crop residue as well. So deep burial of crop residues. So it helps control certain diseases by placing the organism contained in the residue at a depth where there is an oxygen deficiency. So this reduces the population of the disease-causing organism and permits the crop to escape much of the damage. So for this to work, the residue has to be buried deeply enough that it's not pulled back up during seed preparation and cultivation. So burying diseased material is particularly useful against pathogens that produce sclerotia. So those structures I mentioned earlier that can survive in the soil for a very long time with a very little amount of moisture. And those that affect only above ground plant tissue. So pathogens that particularly cause foliar diseases. However, deep full inversion plowing decreases soil health sometimes by burying beneficial organisms that live in the top few inches of soil, the soil program, the soil profile. So things like mycorrhizal fungi or rhizobium, things like that can actually be pushed down. There's other beneficial microorganisms that actually are predators to pathogens or provide other benefits by breaking down organic material. All that is going to be pushed down with the pathogens as well. And unfortunately, usually pathogens are very resilient. So when you're disrupting a soil ecosystem like that, plant pathogens are usually one of the first organisms or set of organisms that are able to recover, sometimes much more quickly than beneficial microorganisms. So that's something to keep in mind as well. So eradication of hosts to reduce level of inoculum. So crop plants remaining from previous seasons, weeds, wild plants can act as hosts, providing a source of inoculum in the new growing season. So for example, wild grasses such as foxtail barley are hosts to the stripe rust fungus, Puccinia striformis. It's also host to a fair number of other rusts as well. I think it's also a host to leaf rust of barley and leaf rust of wheat as well. So many dicotyledonous weeds are infested with root knot nematodes and other nematodes, and certain legume green manure crops can maintain the halo blight organism, which is caused by the bacteria Pseudomonas syringae phaseolicola. Removing the over-seasoning host will actually eliminates or reduces inoculum for the following season. However, the relationship between weeds and crop parasites are often obscure, and many weeds are symptomless carriers of various viruses, so they don't actually show the symptoms the same way that a crop plant does, even though they're housing these viruses. So in other situations, it may be practical to eliminate alternative hosts because they can be it may not be practical to eliminate alternate hosts because they can be commercially valuable crops. So other important inoculum sources are the alternate hosts of many destructive rust fungi. So some rusts aren't actually capable of completing their life cycle without these alternate hosts. And they also allow the rust to undergo sexual recombination, which can produce new races of the rust, which can have much higher virulence or aggressiveness, or could overcome previously resistant plant species. So you could have, say, a crop of wheat that was previously resistant. Because these alternate hosts are present and the rust is able to complete its life cycle, it's actually sexually recombining, and then you end up with a new strain that's actually capable of overcoming that resistance. So the influence of alternative hosts on a rust disease can be highly variable, and the effectiveness of campaigns to eradicate alternate hosts can be unpredictable. So sometimes you get an alternate host that trying to eradicate it is simply impossible. So an example of that would be rust on Christmas trees, and the alternative host is actually ferns. So the chance that you're actually going to eradicate the entire fern population in the province is basically next to nothing. So two important cereal rusts, so Puccinia striformis, stripe rust, and Puccinia carinata, crown rust, actually require alternative hosts. So in the case of Puccinia striformis, it's barberry, which actually has been mostly wiped out in Canada. So we don't see that sexual recombination here. However, crown rust's alternative host is buckthorn, which we most definitely do see. So it is capable of sexual recombination in the country. With volunteer plants, so from a harvested crop are often a means of carrying disease organisms from one crop season to the next. Like I mentioned, so rusts of cereal and spinach constitute an example of this type of disease dissemination. So cultural practices which reduce the rate of spread of disease. So in many cases, cultural practices are directed at improving the growing conditions of plants by providing proper nutrition, moisture, light, and lack of competition from other plants. So they may also be aimed at creating conditions unfavorable for the development of pathogens. Growers' ability to manipulate the environment is somewhat limited in the field, but in greenhouse and storage facilities, high levels of control over environmental parameters can be achieved. So if anybody thinks back to the disease triangle, I don't know if you've seen it before, but it's basically a triangle that shows what's required for disease to occur. So you need a favorable environment, you need a virulent pathogen, and you need a susceptible host. So when we were talking about resistant cultivars, what we're actually doing is eliminating the susceptible host. We're talking about things like burying crop residues, for instance, or burning crop residues. What we're actually doing is we're eliminating the virulent pathogen. In some cases as well, you can alter, what this is talking about is altering that environment so that that environment is less favorable for the proliferation of the pathogen. So if the temperature conditions aren't right, if it's too wet or too dry, for instance, that can all decrease the chance of a pathogen of a disease occurring because you lack that favorable environment. So even if the pathogen is present and you have a susceptible host present, if that environment is not favorable, you won't see disease. So in simple greenhouses, proper spacing of plants and the provision of good ventilation reduces humidity and inhibits infection by pathogens such as botrytis. So in more sophisticated greenhouses, growers frequently lower the relative humidity, especially at night, to dry the surfaces of plants and discourage the spread of pathogens. In the field, more rapid drying of plant surfaces can be achieved by improving air movement through the crop, by sowing or planting at lower densities, orienting rows in the direction of the prevailing wind, and by irrigating in the morning so plants can dry off during the day. So this all alters the favorable environment. Pruning or training procedures that open the canopies of fruit trees to allow light penetration also improve ventilation of the canopy and reduce the incidence of disease. Conversely, practices which allow plants to stay moist for long periods will favor disease development because you have that favorable environment. So things like windbreaks or other structures that interfere with airflow or irrigating late in the day so plants remain wet all night actually encourage disease development. Other aspects of the environment can be manipulated to reduce disease development. To control the spread of fungi that require ultraviolet or near ultraviolet light for sporulation, growers can cover plants with films that block out radiation in these wavelengths to stop the fungi from sporulating. In storage facilities, proper aeration will speed up drying of a product's surface and inhibit germination and infection by the pathogen. So there's a few things that you can do. Rapid cooling after harvesting and storage under conditions of low temperature and appropriate levels of carbon dioxide not only prolong the life of fresh produce, but will also inhibit disease development as well. So next one, tillage practices. So tillage incorporates various types of organic matter, including crop residues, manure, green manure, volunteer crop plants, and weeds into the soil. It tends to have indirect effects on the spread of plant pathogens, although some forms of inoculum can be widely dispersed by implements. It reduces populations of weeds and volunteer crop plants, however, that harbor pathogens between crops, so that can be beneficial. And it also buries plant pathogens from the soil top layer, into deeper layers of the soil where they cause less or no disease. So we mentioned that before when we were talking about bearing residues. Although, like I mentioned before, that can potentially have detrimental effects as well, because you're also bearing those beneficial microorganisms. Practices involved in the preparation of seed beds can greatly modify physical properties of soils, such as moisture characteristics, bulk density, aeration, and temperature profiles, which in turn influence the incidence of disease. So things like planting on a raised bed can be helpful in preventing certain diseases such as southern blight and particular wilt diseases. This practice is advisable when growing legume crops such as soybean and when growing vegetables in tight, poorly drained soils. So forming the soils into hills, ridges, or raised beds provides better drainage and irrigation. Tillage may also influence nutrient release mechanisms, and the total effect is often seen as increased crop vigor. Healthy plants may be more resistant to some pathogens, but the more humid microclimate within the crop can be conducive to the spread of other pathogens. So successive tillage operations can reduce inoculum levels of some pathogens through exposure of the inoculum to desiccation by the sun. So basically drying out. So you're moving that soil around. You may potentially expose some of the inoculum, which can potentially dry out. Minimum and no tillage. So minimum tillage is a method of planting crops that involves no seedbed preparation other than opening the soil to place the seeds at the intended depth. So usually the soil is not cultivated during crop production, and chemicals are used for weed control. So this reduces injury to the roots of crop plants caused by mechanical tillage or hand weeding, reducing the opportunities for opportunistic pathogens to infect. It also reduces the spread of pathogens by tillage practices. So one thing I hadn't mentioned before about some tillage practices is when you're dealing with plant pathogens, it's always preferable for things to be dispersed vertically. So you're bearing those pathogens deeper. It's always not beneficial for that material to be dispersed, say, horizontally, because what you're actually doing is you're spreading the pathogen inoculum around. So in no tillage systems, crop residues are left on the surface of the soil. The residues may become a food source for pathogens or alter the physical environment occupied by the host and its pathogens. So the effect of tillage practices on the soil microflora is often overlooked. Many of these organisms are competitors or antagonists of soil-borne pathogens. Minimum tillage practices are thought to promote greater microbial antagonism in the vicinity of roots than normal cultivation practices. So the incorporation of organic matter, so things like green manure crops, is known to reduce the incidence of some diseases. So plant residues intensify the microbial activity of the soil, which may result in the formation of fungitoxic compounds, which may inhibit pathogens. So adjust crop planting to disrupt pest habitat. So crop planting can be adjusted both in space and time to reduce the development of large pest populations. So alternating the time of planting to avoid high levels of pathogen inoculum or conditions conducive for development of reticular disease can lead to reduced severity of some crop diseases. So the best thing to look at when we were talking about the disease triangle in a favorable environment is to avoid the environment where that pathogen can proliferate entirely. So if you're able to plant before that pathogen actually becomes a problem and the plants can reach a certain level, a certain growth stage, maybe where they're able to fend off more, they're more able to fend off the pathogen, or they're actually avoiding the part of their life cycle where they're most susceptible. That's about the most beneficial thing you can do to avoid pathogen infection. So is actually missing that window where the pathogen can infect entirely. So for example, like early plant fields of soybean may have greater incidence of seedling bites caused by Fusarium solani and Pythium if planted early in cool and wet soils. So the incidence of these two diseases can be reduced by delaying planting until the soil warms up. So many crop plants tend to be more susceptible to attack by various pathogens at certain stages of their development. Like I was saying, so changing the usual sowing time of crop can exploit weather conditions which are unfavorable for the spread of the pathogen or their vectors. So things like insects that spread the pathogen and reduce crop losses. So the next thing is crop density. Crop density can exert considerable influence over disease due to the ease with which the pathogen inoculum can be transferred between closely spaced plants. In closely planted crops, temperatures are more uniform, humidity can be higher, and foliage is wetter for longer periods of day. All of this stuff provides a favorable condition for pathogen infection and subsequent development. So diseases like downy mildew and white mold are greatly affected by high humidity. So they proliferate much more rapidly under humid conditions. So like I said, crop density can influence disease development. Generally, as a rule, as the density of a crop increases, the incidence of the disease also increases. So for example, in the 1950s, due to an increased demand for seedlings, nurseries greatly expanded in size and increased the density of seedlings in their nursery beds. What ended up happening was they were seeing a lot more crowding of the seedlings, and they ended up getting serious epidemics of damping off caused by fungi like Pythium, Fusarium, Rhizoctonia, and Cylindrocladium. So there are a number of reasons why high plant densities increase disease incidence. Most are associated with the ease of transferring inoculum from one plant to another when the plants are close together. So for example, the distance pathogens or their vectors have to travel from one host to the next is greatly reduced. You can end up with splash dispersal of inoculum, which is much easier. So you have an infected plant, the rain drops, and it actually splashes the inoculum from one plant to the other. And there's also increased contact between the leaves or roots of neighboring plants. So you have an infected plant and a non-infected plant, and the leaves are actually touching, or the roots are touching, allowing the pathogen to move essentially from one plant to the other. In addition, with denser plantings, more plants may be wounded during cultivation, creating more opportunities for weak pathogens to infect. And then finally, the microenvironment within the crop is altered. So temperatures are more uniform, the relative humidity increases, and leaves usually stay wet longer after rain or dew because you've got that closed kind of canopy. All these conditions will favor the development of disease. So farmers can manipulate plant densities by varying sowing or planting rates, and may use increased rates to compensate for expected crop losses from pathogens. Crop density can also be manipulated by pruning, thinning, trellising, fertilization, water management, staking and harvesting plants or plant parts, depending on the type of crop. And all these practices are designed to prevent foliar root contact, which will reduce the spread of pathogens. So depth of sowing. So deeper seeding may promote germination, but it also lengthens the usually susceptible pre-emergent seedling phase. So the seedlings, they're slower growing initially, and they're more prone to be attacked by seedling diseases caused by Fusarium and Rhizoctonia, smuts as well. And it's more serious if plants are seeded very deeply. Similarly, potato seed pieces are more readily attacked by Rhizoctonia if planted too deeply as well. So next, we're going to talk about intercropping. So the incidence of disease is often less in mixed plantings than in monocultures because the distance between similar plants is greater than more intensive growing systems. So it's less likely that spores or vectors of pathogens will successfully move from one host to the other. The intervening plants pose physical barriers to the dissemination of aerial pathogens, so their release or their vectors, such as insects. Cover crops, so disease suppressive cover crop rotations may provide an additional tool for managing soil-borne diseases. The effect, however, is highly variable, and it differs between locations, between years, as well as the cover crop used and the cover crop by pathogen by environment interaction. So like I said before, thinking about that disease triangle, we're looking for cover crops that are not susceptible to the pathogen. They don't create a favorable environment for the pathogen. And there's certain cover crops too that are naturally resistant to certain pathogens, or they create environments that actually inhibit pathogen growth. So whether that's altering the physical characteristics of the soil, so that the pathogens it's not conducive for pathogen growth, or it's more conducive for the growth of antagonistic microbes that could attack the pathogens. Some of these cover crops can actually produce antifungal or antimicrobial chemicals that could inhibit certain plant pathogens as well. Mulching. So the application of a covering layer of material to the soil surface is a commonly used cultural practice, especially in horticulture. Natural materials used for mulching include cereal, straw, and stalks, crop debris, sawdust, leaves, grass, compost, and manure. So crop residues generally enhance competition among soil microorganisms for nitrogen, carbon, or both, resulting in fewer problems with soil-borne pathogens. However, in some cases, the incidence and severity of disease has increased with mulching, perhaps because the microclimate within the system has changed in ways which is favorable to the plant pathogen. So organic mulches reduce loss of water from soil, increase infiltration and absorption of water, smother weeds, lower soil temperature, sometimes by as much as 5 degrees, enrich soil by supplying nutrients and organic matter, and protect seedlings against the impact of rain, hail, and wind. All these factors may influence disease development. Spread of soil-borne disease is reduced because a layer of mulch on the soil surface reduces water splash dissemination of bacterial and fungal spores and prevents leaves, flowers, or fruits from directly contacting the soil and disease spores in the soil. So plant nutrition. So a properly nourished plant is able to withstand or tolerate the attack of pathogens much better than a plant that has nutrient deficiencies or has been excessively fertilized. Fertilizer use has some bearing on development of certain diseases. It differs with each crop and each disease, but in general, nitrogen out of balance with other nutrients enhances foliar disease development and predisposes some plants to other diseases. So soil nutrient status may influence the susceptibility of plants to attack by pathogens. So healthy, vigorous plants should have a greater chance of withstanding attacks by pathogens, although this isn't always the case. The same conditions that favor the growth of plants may also encourage development of biotrophic pathogens, so pathogens that survive on living material. So for example, many viral diseases of crop plants are promoted by fertilizer applications. So when applying fertilizers, it's best to think about the 4 R's. Right rate, particularly, so you don't want to be applying not enough new fertilizer, because then your plants are going to be small and weak and susceptible to pathogen attack. But you also don't want to be applying excess nutrients, because then you could potentially be feeding the pathogen population. Or you look at your plants are producing too much vegetative material, which may be prone to pathogen attack. So nitrogenous fertilizers. So application of nitrogenous fertilizers are commonly thought to predispose plants to disease. Nitrogen applications tend to delay crop maturity, like I said, by prolonging vegetative development, which can increase the risk of infection. And in contrast, the stimulation of vegetative growth may allow plants to outgrow an infection and compensate for any damage as well. So high nitrogen levels are also thought to influence the production of host metabolites, which can either inhibit or promote infections by various pathogens. So a lot of these fertilizer recommendations for reducing plant pathogen populations are very complex. So you see in the literature a lot of like, if it's detrimental to have too high fertilizer levels because of this, it's also detrimental to have too low fertilizer levels. So like I said before, having the right rate is incredibly important. So phosphorus fertilizers, the effect of phosphorus fertilizers on disease incidence are not well understood, but the effects of phosphorus fertilizers are attributed to correction of phosphorus deficiency in the soil, which can result in healthier plants more able to resist attack by pathogens. In addition, phosphorus hastens the maturity of the crop, so young susceptible tissues are not exposed to the inoculum of obligate parasites for a long period of time. Potassium fertilizers, so adequate potassium levels inhibit the development of a wide range of pathogens, including fungi and bacteria, as well as various viruses and nematodes. And application of potassium fertilizers also seem to reduce some of the disadvantages of excessive nitrogen application. So the effect depends on the nutrient status of the soil, the rate of application, and the buffering effect of potassium on other elements, especially nitrogen. So irrigation management. Irrigation management is clearly an important factor when it comes to disease control. So regardless of the irrigation method a grower chooses, timing and duration of irrigation should satisfy crop water requirements without allowing for excess water. So irrigation can have a major influence on the spread of some pathogens and on disease development. Irrigation applied during dry seasons means the spores of pathogens are not exposed to desiccation during periods of drought, so they're going to stay viable, which can cause the level of inoculum to increase. In addition, irrigation water may contain spores of pathogens, which carries from one place to another unless it's carefully treated before use. Overhead watering can promote disease by increasing the period of time a layer of free moisture remains on the leaf surface. The longer leaf wetness period increases the likelihood that sufficient time will be available for fungal spores to germinate, form infection structures, and penetrate the plant surface. Irrigated crops may become a green island as well in an otherwise dry environment and attract insect vectors of virus diseases. In such cases, it's often better to delay sowing an irrigated crop for some time after other vegetation has dried up and the vector population has been reduced by desiccation. Irrigation can be used as a tool to reduce the level of inoculum and to retard development. Alternatively, drying and re-wetting soil encourages the activity of microorganisms that may destroy sclerotia. So flood furrow and overhead irrigation can facilitate the spread of pathogens. Flood irrigation can spread soil-borne inoculum all over an area, while furrow irrigation disperses inoculum along rows. The action of overhead irrigation systems wash inoculum out of the air and facilitate the spread of pathogens that rely on water splash for dispersal. Many important foliar and fruit pathogens, such as Phytophthora infestans, so late blight and Alternaria solani, form their spores at night and release them during the day. So overhead irrigation in the early part of the day washes these spores from the air and splashes them about. If overhead irrigation is delayed until the evening or early night, spores of late blight dry out on the plant and can infect, although spores of Alternaria are actually resistant to drying out and can survive until a dew forms at night. So the timing of overhead watering has little influence on this disease. So knowing what kind of disease and its conditions you're dealing with are also important as well. Trickle or drip irrigation, developed in response to the need to conserve water, supplies water directly to the root zones of individual plants, and the rate of application is insufficient to disperse pathogens. So it's actually not enough to actually move water to actually move pathogen populations or pathogen spores around. As well, drip irrigation produces a mosaic of soil moisture conditions rather than uniformly moist conditions, which probably inhibits the spread of root pathogens. Here, we just have a couple more. So roguing of diseased plants as they appear is often an effective method in helping reduce the spread of a destructive disease. Virus diseases of stone fruits and bacterial wilts of cucurbits are examples where roguing is worthy of consideration. So these cultural control practices have been found to be economically feasible in reducing disease loss. Growers should properly identify the disease that limit production and use a variety of controls in combination. Finally, we have sanitation and preventing the buildup of pathogens. So wash soil off farm equipment, including brushing off soil particles from shoes. These practices are especially important to prevent movement of soil-borne pathogens, such as white mold, Phytophthora, verticillium, and different species of Fusarium. Power washer is an important piece of equipment in the battle against these diseases as well. So that's what I have for the presentation.
Thank you, Dustin. That was fascinating. Before you stop sharing your screen, one of the questions was about the intercropping picture you had. I think it was a brassica and a grass.
Oh, yeah.
Just asking about what species they were, or what crops.
The main purpose is the grass is actually sowed in the center, and the brassica is on both sides. So it's actually preventing splash from occurring over the entire crop, because that grass is actually growing up taller, so it's actually acting as a barrier to prevent splash dispersal across that entire row. I think that's the main purpose.
Okay, thank you. And if you want to stop sharing your screen, I will share the CCA QR code just before we finish.
There we go.
Okay. And there was another question about if you could talk about spraying in the evening or at night. Would that be a risk of disease?
Oh, can you say that again?
Is it a risk of disease to spray in the evening or at night or during the day?
Oh, most definitely. Yeah, most definitely. Yeah.
Okay. I had another question about, so you talked about pathogen loads on seeds.
Yeah.
If you're getting treated seed, say I'm a broccoli grower and I order some treated seed, should I expect the seed to be pathogen free, or will there still be a pathogen load on the seed even though it's been treated?
Yeah, so if it's been treated, I wouldn't expect there to be pathogen load on the seed, because that treatment should essentially, it's not designed to kill the pathogen necessarily. Like most products aren't. What it is it's preventative. So at most, what it's going to do is it's going to suppress any pathogens that are present, and it usually suppresses those pathogens for a certain amount of time, which allows the seed to germinate and reach a certain growth stage that it's no longer susceptible to those pathogens. That's what it's doing essentially. So even if the pathogens, I guess theoretically they could be present on the seed surface, but they're going to be inhibited, severely inhibited. So you're not going to see any symptoms develop, because that's the purpose, right? Is it suppresses that pathogen until, because it's suppressing pathogens. The treatment is designed to probably specifically to suppress pathogens that are virulent to that crop in its seedling stage, right? That's what it's intended to do. So by the time the effects of that treatment wear off, that crop should be outside of the range of that growth stage to be capable of being infected. Yeah. The other thing I guess I should have extrapolated maybe on that irrigating at night thing is that the main detriment to that is that you have no light and you have colder temperatures, right? So that wetness is going to be on that leaf surface for an extended amount of time, which is going to cause the pathogen to proliferate. You may see a reduction potentially if it's cold enough, right? It could slow down the aggressiveness of that pathogen, but it would always be preferable to irrigate, say, early in the morning, where you're able to get that drying effect, right? Through like you're able to get the sunlight and that drying effect, because you want to get that water off of the leaf surfaces as quickly as possible, because the pathogen actually most pathogens, fungal pathogens particularly, they require a certain amount of water to actually run the fungal apparatus, so that the actual organ that allows for infection to occur. So they produce a structure, a lot of pathogens called an aprosoria. And the aprosoria, what it does is it actually punches through the leaf cuticle. Without enough water, it's not actually able to the aprosoria is not actually capable of functioning. So what you're actually looking to do is try to reduce the amount of time that those leaves stay wet, as much as you possibly can. Like that's the overarching goal when you're dealing with irrigation, when you're thinking about plant pathogen infection.
Great. Okay. I think there are no more questions in the chat, and we're just over time, so we'll probably call it a day there. And that's a good take-home message. Don't let the aprosorias establish. So
yeah, if you know what an aprosoria is.
Yeah, thank you everyone for joining us for the last session of Getting into the Weeds. It's been great, and we will see you next year.